I think that's all the announcements I've got, so I will hand it over to Marianne to introduce Holly.
Hi. Yeah, it's my pleasure to introduce Holly Johnson. Holly is an undergraduate in the Department of Botany at UW-Madison and soon to be graduate student. She is our winner of the John Thompson Research Award from the Botanical Club of Wisconsin. She's also a Madison local, so she grew up here in Madison. And Holly has been working in the Wisconsin State with me for about 2 years now. Um, One of her very first projects was entering a whole bunch of moss specimens that we found that had actually been collected by L.S. Cheney between 1880 and 1932. And so she just started entering the data into our database and also transferring these specimens from this really old, nasty newspaper into um, archival packets. And so this could have just been a really kind of mundane, straightforward project, but Holly has really transformed it into something else. So she just became really fascinated both with Ellis Cheney and also with sphagnum mosses because a large portion, several hundred of these mosses were sphagnum or peat mosses. So she became very interested in peat mosses and also what this new data that she was entering into the computer would tell us about the distribution of sphagnum throughout Wisconsin. And then she also became very interested in the man himself, the collector Ellis Cheney. And so that's what she's going to be talking to you about today. And so with that, I'll let Holly take it over. Thanks, Holly.
Hi everyone, can you hear me all right? Thumbs up. All right, sounds good. So I just want to start by saying thank you so much to everyone who came out today, and thank you so much for having me here. I am so excited to share this project with you, not just because it's something that's so special to me and because it's something that I've learned so much from, but also because I think that you guys are exactly the group of people who will find meaning and interest from it as well. Yep. So I want to start with a little bit about me, further than what Marianne shared, just so that I'm not a complete stranger talking at you for the next 40 minutes. So as Marianne said, I'm a student of botany here at UW-Madison. I'm a senior, so I'll be graduating in exactly a month and 2 days from now. It's exciting. I'm an employee and student researcher at the Wisconsin State Herbarium, working with the bryophytes, which is what got me involved with this project. I'm also part of the Students in Water in Wisconsin's Peatland Plant Communities Research Group, which is also spearheaded by Marianne Feist. But outside the herbarium, I am also an undergraduate researcher at the UW Plant Path Department, and there I study the genetic basis of necrotrophic fungal pathogenesis and Sclerotinia sclerotium, and I'll be beginning my master's here studying botany in the fall. Outside of school, I've worked jobs at the UW Arboretum and in the native plant garden under the fabulous Dr. Susan Carpenter, and as well as at Olbrich Botanical Gardens. And outside of school and work, I'm a metalsmith, so I make a lot of my jewelry, and I enjoy kayaking and tending to the dozens of houseplants that I have around my apartment. So, as a little summary of what we will be talking about today, I want to start off by telling you a bit about Leland S. Cheney that Marianne introduced, and how the integration of his bryophytes into the Wisconsin State Herbarium led to the research questions that allowed me to begin on this project that I will talk about in section 2. And then in the third and final section, I would like to do a brief overview of sphagnum moss microscopy, just because this is something that actually ended up taking the majority of the time that I spent on this project. It's something that is incredibly intensive, but something that I've grown to really love over the past year, and something that hopefully you'll grow to love too by the end of this presentation, but that might be wishful thinking, so stay tuned. And with that, I would like to get started on section 1, the rediscovery of Leland S. Cheney's bryophytes. So in 2016, during some renovations of a few offices on the third floor of Burge Hall, which is where the UW Botany Department as well as the herbarium are housed at UW, this led staff to come upon dozens of boxes of what added up to be over 2,000 unprocessed moss specimens. Um, and so this was exciting. A lot of these specimens, the majority of them were labeled to have been collected by a Leland S. Cheney, who was a botanist that was already significantly represented in the herbarium for his contributions to the collections of vascular flora across Wisconsin, in the early 1900s and late 1800s. There were also a few other collectors represented amongst these mosses, such as Stunson Allen. These are a few botanists that did a significant survey of the mosses of Isle Royale. I think it was in 1905, but the majority of these specimens were from Leland S. Cheney. And so many of these mosses were neatly packaged up in cute little packets that were labeled collected and identified by Leland S. Cheney, all nice with a labeled species name. But a lot of them were just wrapped up in these temporary newspaper packets that you see here on the left picture. And so some of them had locality information associated with them, as you can see on the right, but some of them had nothing with them. And so these were really fun to look through as we were databasing these specimens. All of these newspapers, they were from, like 1920s. And so I came to enjoy looking through them. And did you know that in 1925, you could buy a prime rib for about 20 cents a pound, and you could buy an all-inclusive trip to London from Chicago for about $100. So it seems like a steal to me. Fast forward about 8 years, and I was hired on at the Wisconsin State Herbarium in 2024 to assist with the digitization and the restoration of these specimens into the herbarium. And so this started with a lot of transferring those mosses that were wrapped up in the newspapers to fresh packets that were made from acid-free paper. And so this is important in the herbarium to preserve the integrity of the specimens and ensure that they aren't degrading in normal paper over time. And so I folded a lot of those acid-free packets, and I also folded up tiny little slips that fit inside to those pre-existing packets that I talked about that were labeled with the collected and identified by Leland S. Cheney, because we thought that was an important part of history to preserve in the herbarium, even though it didn't, you know, it wasn't acid-free at the time. And so after we made all of those transfers, the next task was to database all of these specimens. And so I like to think of this job kind of like an endless puzzle. There are just endless places that you need to look in order to find the locality information that matches up with each of these specimens. This Cheney collected like 17,000 plants, I think, and that's just over the course of so many different collectors' logs. And so it was really fun to really get to know the chaos that was his collecting over the years, and to go through these collector's logs. And I really fell in love with the process of it all. And so um, I entered all of this locality information into the database. And finally, the third step to this is making sure that the name that was assigned to the moss at the time that it was identified, so, like 130 to 100 years ago, is still taxonomically relevant. And as you can imagine, a lot of the times it's not true. So I have to do research in order to figure out what the most recent accepted name is for each of these specimens. And so, with that, I'd like to tell you a little bit about the man, the myth, the legend, Leland Sterling Cheney himself. So, Cheney was born in Essex, Ohio, on December 4th, 1858. He then went on to become a student at UW-Madison from 1889 to 91, and then after that became an instructor and associate professor of pharmacological botany at UW from 1892 to 1903. And so it's interesting. I was talking about those newspapers that he used to fold up those specimens, but he would also recycle the homework assignments that his students at the time would hand in, his botany homework assignments to wrap up his mosses. And so I had a really fun time looking through, homework that was assigned to botany students like me in the early 1900s and comparing it to my 21st century botany homework. And during the time he was an instructor at UW, he was also the curator of the Wisconsin State Herbarium until 1903, when, owing to ill health, he retired from UW and relocated to Barron, Wisconsin, where he served as the Barron County School Superintendent. Um, but he didn't lose his love for collecting plants. He was collecting plants all the way until the end of his life, and he passed in 1938. And so Cheney was one of Wisconsin's pioneer plant collectors. He collected all across the state from 1893 to the 1930s. He is known for his significant contributions to the vascular flora of Wisconsin, of which he collected a lot on these really epic trips that he would take the train across the state to the general area where he wanted to collect. And then he wrote about how he commissioned people to build him a canoe at the top of the site where he wanted to start collecting. And then he would spend months in this canoe gathering thousands of specimens and then return home to the herbarium with those. Um, but he really paid special attention to the bryophytes of Wisconsin as well, and especially during the later years of his career, he really ramped up the collections of, sphagnum, and not just sphagnum, but just, like dozens of species of bryophytes that are not very heavily represented across Wisconsin from anyone else but him. And so this leads me to my project because, as I worked over the past 2 years to integrate these collections into the herbarium, my boss, Marianne Feist, and I, who many of you love and know, we began to wonder, what are the states of these sites that he collected at today? Are they still in existence, or are they in Ace Hardware parking lot, which is a true story? And if they still exist, are they still a wetland? If it was sphagnum that was collected there, if it's still a wetland, is the sphagnum present? And if there's still sphagnum present, finally, how has the composition of those species changed over the past century to 130 years ago? And so with that, that leads me to section 2, where my project investigates a century of change in Wisconsin sphagnum. So for those of you who are familiar with plant collecting for an herbarium, maybe that's through the Botanical Club's forays, or maybe you collect independently, maybe you used to work in an herbaria, um, you're probably familiar with the importance of providing detailed and accurate locality information with every single one of the plant specimens that you collect, right? So here in this picture here, you can see a sphagnum specimen that is wrapped up in that newspaper. And this was collected in 1928. So this was a little bit later in Cheney's career. And after 12,848 collections, Cheney finally learned the importance of including detailed locality information with every single plant. So this reads, near the edge of a small marsh in section 29, Township Newark, Rocky County, Wisconsin. And with this detailed locality information, I was able to track down this exact site and give it a survey and check to see if sphagnum was still there. And unfortunately, sphagnum is no longer present there. But that's not my point right now. My point is that if you take nothing else away from this presentation, have it be the importance of those locality descriptors, because earlier in his career, unfortunately, a lot of his descriptions were just, like Madison or Lake Mendota. And, you know, for the sake of, some undergrad who might be scouring over your specimens 120 years in the future, wondering where on earth in Lake Mendota did Cheney find sphagnum, it's useful to include, like GPS coordinates with every single specimen. And so that takes me to where my project begins in Barron County. And if you'll remember, Cheney retired here in 1904 after his time as an instructor at UW-Madison. And so because he retired here, a lot of his later collections are concentrated around here in Barron and the surrounding area. And because they were later in his years, he had TRS coordinates associated with many of these specimens, or with many of these sites. You can see here a lot of them are concentrated around this small stream called Quadra Creek. And so each of these little blue bookmarks marks one site that Cheney collected sphagnum at along Quadra Creek. And so we will start with sections 18 and sections 20, which are the northernmost sites along Quadra Creek. And for historical accuracy purposes, we decided that it was important to collect via canoe, just like Cheney did. And so here are some pictures from our collecting trip in section 20. Um, on the top right, you can see me posing with something green in my hand. You might expect it to be a fish, the way I'm so proudly, holding it up in the pose that I have, but it's just a ball of sphagnum because we ended up finding sphagnum here again. So in the top left, you'll see me smiling. Don't be fooled. I'm smiling through the pain. I'm smiling in an alder thicket, which is where I'm kidding. I'm really excited because we found the sphagnum that you'll see in the bottom right hand corner. And so I think when a lot of people think of sphagnum, they think of those characteristic hummocks and large lawns that you'll see across an open bog. But the sphagnum that we found in section 20 of Barron County, it wasn't like that. It was much more patchy. They were tiny little patches of it. Much of them were on the base of alder mounds, just like this pictured here. I found a little bit in some cattails at the bottom of it was like pretty much just in the water, and then cattails, and then there's some sphagnum at the base. Um, and so if you look at this Venn diagram here, comparing the species that were collected in 93 to 2025, you'll see that it was puzzling to me. Sphagnum was present. We found Sphagnum gergensonii, squirosum, and Sphagnum fimbriatum, but there was absolutely no overlap with any of the species that were collected in 1893. But what was further puzzling to me is that these species that we collected in 2025, these are all generally characteristic species of more minerotrophic habitats. So Gergensonii, squirosum, fimbriatum, these generally grow in minerotrophic habitats, indicating a preference towards less nutrient flow. And so a lot of these species that you'll see on the left hand, collected in 1893, these are Sphagnum capillifolium and Sphagnum fuscum. These are generally more characteristic of ombrotrophic habitats, which means this would indicate a preference towards less nutrient flow and just instead fed by rainwater. And so you'll see here, if you compare these two data sets and use the Jacquard index value, it comes out to be 0, indicating 100% species turnover over the past century. And so this is section 18, that northernmost section that we were talking about. In 1931, Cheney collected Sphagnum fimbriatum. This was a bit later than his earlier collections. The section 20 was 1893. But in 1931, he collected fimbriatum, and in 2025, I searched this quarter section high and low, far and wide, and there was no sphagnum habitat to be found. I was really hopeful, but I didn't find anything. I can pretty confidently say there's no longer sphagnum present there. And so that brings us to section 29. And unfortunately, section 29 was smack dab in the middle of a private golf course. So the closest that I got to section 29 was this picture here of it. And I got to stand on a bridge and look out over it and really think about it a bit. I didn't get to do as in-depth of a survey as I would have liked to do. But just looking out over this to me does not scream sphagnum habitat. I cannot say for certainty that it's not, but it does not resemble what Cheney described the site to be. He indicated the presence of some tamarack trees here. I do not see those at all, and this is where his locality led me to, and it does not look like he described. So with that, I made the executive decision to label this as no longer sphagnum habitat. And so moving on to section 32. In 1931, Cheney collected Sphagnum divinum and Sphagnum warnsorfii from this site, and in 2025, do not be fooled. This looks like it's just over the hill of a nice green hill, but that's private property. So I had to walk through quite a bit to get here, and I can say with 100% certainty, this is no longer sphagnum habitat. It is completely overtaken by invasive species and reeds, and there is no place that I would expect sphagnum to be here, and I spent quite a long time here with no luck. Now moving on to section 6, the southernmost sites in Barron, we found sphagnum again. So Marianne and I visited 2 sites here in section 6, and you'll see here one of them is more of a cattail marsh. One of them, I didn't put a picture here, one of them was more of a marshy sedge meadow situation, and at both of them there was sphagnum present. You can see Marianne there and some thick typha. Um, but again, in 2025, we collected Sphagnum centralis, Subsecundum, Squarosum, and Gergensonia. These are species more characteristic of minerotrophic habitats. In 1928, Warnsdorfii was collected, and Warnsdorfii is interesting. This is not necessarily sphagnum you would find in an ombrotrophic bog, but it is something that is a little bit more of a calciphile. Actually, it has a pretty high calcium tolerance, and so you'll generally find sphagnum warnsdorfii in a poor fen of such. But this is definitely not a poor fen, and there's definitely not sphagnum warnsdorfii there anymore. And thus, the Jacquard index value comes out to be 0 once again, even though there was sphagnum present. And so, over the course of my sphagnum surveys over the past year, I did not just limit myself to these sites that were collected at by Cheney. And so, on this particular trip, Marion and I made a visit to a very lovely sedge meadow that was pretty high quality. And at that site, we uncovered a lot of those species that Cheney had reported collecting at those sites that we didn't find there. And so I think it's really interesting if you take, on a site by site basis, a lot of those sphagnum species have been lost over the past 130 years. But if you take a step back and you look at the county as a whole, those species are still present a lot of the time, but they are just present, in higher quality habitats. So a lot of those species in the middle were species that Marianne and I found at this higher quality sedge meadow. And that's not to say that Sphagnum capillifolium, Palustri, and Wolffium and Papillosum that were collected by Cheney between 1893 and 1931 are no longer present at all in Barron County, because they certainly didn't do an exhaustive survey of the entire county. But yeah. And so when during our trip to Barron, we ended up finding Cheney's grave, and we paid it a visit and paid our respects. And you might notice a little bundle of something green in my hand, and that is, in fact, a little collection of sphagnum moss from our trip along Quadrier Creek. And don't you fret, we left it at that base of his grave site in a thank you to him for all that he's done. And so here is a map of all of the sites that we visited along Quadrier Creek. You'll see that of the 6 sites that we visited, 4 of them had no sphagnum present at them. 2 of them had sphagnum present, but there was no overlap between the species that were collected by Cheney and collected by us. And so that brings me to the second trip of my project in Bayfield County. And I visited 4 sites in Bayfield, 3 of which were exactly where I know that Cheney collected. And so we'll start with Port Wing Boreal Forest. Port Wing Boreal Forest was lovely. It was very large and expansive. It was an open bog, sedge meadow wetland complex. Um, there was sphagnum certainly present there in abundance. There were a lot of Sarracenia, Drosera, leatherleaf, Eriophorum, a lot of, you can see some tamarack back in the back left side of that larger habitat photo there. A lot of those vascular associates that you would typically expect to find with sphagnum at a higher quality habitat, unlike those that we found in Barron. So this was a bit of a change. You can see me, I'm holding a little bundle of what I believe is Sphagnum mesorecum. And so this was interesting. There is quite a spread between the species that were found in 1897 by Cheney, in 2025 by us, and that were shared between the two. And so that comes out to be a Jacquard index value of 0.33. And you'll find that this pattern is preserved across our next site. So a couple miles up at Bark Bay Slough State Natural Area. This was more of a coastal bog situation. And so a lot of those vascular associates that I had just listed earlier, I was excited to find this little bumblebee in the corner. It's one that you'll only find in the north. It's not found in southern Wisconsin, so I was excited to find that. that on the pictured on the top there is Sphagnum subsecundum, and we ran out of the bags, the paper bags that we typically use to collect sphagnum in the field, so I had to resort to using coffee filters that we had at the place that we were staying. And so again, this distribution of species is pretty spread between species that were strictly collected by Cheney, strictly collected by me, and shared between the two data sets with a Jakarta index value of 0.27. And so what I think is what happened here is that these sites were so large and so expansive and so diverse and intact that I thought that the locality descriptions that Cheney had given me were specific enough to track down exactly where he had collected, like in Barron. But because these sites are so expansive, I don't think that I was able to locate exactly where Cheney had collected at. As much as I would have loved to, I did not have the time or resources to do an exhaustive survey of the entirety of all of the sphagnum across both of these state natural areas in the weekend that I was there. But that's not to say I think that these collections that I made here are still incredibly valuable. I made some collections of a few species that are not very common across Wisconsin. And so for that, I'd like to think that's really valuable information for context of their distribution across North America. And I also think it's an important context for a snapshot of the diversity of sphagnum that we found at each of these state natural areas, because not many other people have collected here. And so I'll move on to the last site here in Bayfield County, which is Friendly Valley Beach. And this site was a little hard to access. It was really overgrown. I think this picture makes it look a little bit more polite than it actually was. It was really overgrown with like canary grass and typha. Um, and so I ended up collecting one specimen, and that ended up being Sphagnum teres. But in 1896, Cheney collected Sphagnum gergensonii. And again, this might just be a matter of I did not make it to the exact same spot where he collected gergensonii, or maybe it was lost over time. But that was an issue that was presented in Bayfield that was not necessarily present in Barron. And so again, if you zoom out and look at Bayfield County as a whole, there were a lot that were shared between our two data sets, a lot that I actually found that Cheney didn't find, and a couple that Cheney found that I didn't necessarily find. And I don't think it's necessarily that they've been lost over time, though. Like I was more confident that they had been in Barron County. And so now moving on to our first site in southern Wisconsin. This one is in Waukesha County. And I was really excited when I hiked out along some railroad tracks and I finally spotted a little grove of tamarack trees off in the distance. The locality description being in Tamarack Swamp, and then he provided the TRS units. So I was pretty confident that that was exactly the site where he had collected because, yeah. And so I knew that was a site where he had collected, but what I did not know was that I was then going to have to traverse through about a third of a mile of really, really, really dense cattails and then a really, really, really long alder thicket site. So that was really fun. And then when I made it there, most of these tamarack trees ended up deceased. And so I'm not sure if that was the doing of the eastern tamarack beetle, or if it was just more of a successional shift away from a habitat where tamarack can thrive anymore. But what I can confidently say is that this is no longer sphagnum habitat. In 1925, Cheney collected Sphagnum divinum, Sphagnum palustri, and squarosum. Um, and you might be thinking, hey, you walked through an alder thicket and you found some species of sphagnum amongst the alder in Barron County, but Sphagnum divinum, palustri, and squaresome are not characteristic species of alder thicket, and I did look there and I found nothing. I spent the better part of an afternoon here, and I moved on with it. Comfortable saying with confidence that this is no longer sphagnum habitat. And so now moving on to the last site in that I'll cover in Rock County. Wisconsin. The locality description was near edge of small marsh in section 29, Township Newark, Rock County, Wisconsin. And this might sound familiar because this is the description from the example earlier when we talked about the importance of locality descriptions. And so I was really looking forward to this site, and I was really hopeful that I would find sphagnum here. But unfortunately, I again checked far and wide, high and low. I I really tried hard to find sphagnum here, but I did not find anything, except for a lot of dead animals. So that kind of freaked me out. And so there are a few sites that I would like to visit into the future. Some of these I'm not too confident that there will be sphagnum at, but I still want to pay a visit to them. One of which being one in Dane County, the locality description for that being at edge of a small pond, 1 mile east of Lake Monona. And I've deduced that I'm pretty sure this is probably the Tamarack Grove at the Aldo Leopold Nature Center. So I'd like to go pay a visit there, not because I think that sphagnum is there. I don't think it is, but just to see, where it once was. There's a site in Waukesha County, Huckleberry Marsh near Whale Sanitarium. This was one that was a really significant site for Cheney. He collected a lot of sphagnum here, a lot of different species, and so I would really like to visit there someday. The problem is that it's on private property, so I'm gonna have to figure out a way to let somebody beg somebody to let me in there. There's a site in Burnett County, the locality description being in swamp near Webb Lake PO. I think that stands for post office. Um, I have high hopes for sphagnum present there. And another one in Dane County, and maybe you guys can give me a help with this. The locality description of this one is margin of a marshy meadow about 1 mile north of Mendota Station, rare. And so this was a little bit earlier in Cheney's career, and he said rare to Sphagnum gorgonsonii, which is definitely not rare in Wisconsin. Maybe it is in Madison. Maybe that's what he meant. But what I'm not sure about is this Mendota Station. I've done some research online to try and figure out what he might be referring to, and there's like 6 different Mendota stations he might be talking about. But if you guys have an idea of a Mendota Station where a mile north there might be some sphagnum habitat, let me know. And so here is a map of all of the sites that I resurveyed over the past year. You'll notice that all the sites in southern Wisconsin, I did not find any sphagnum there. I did put a label on the sphagnum sites that he labeled as Mendota, because there's definitely no sphagnum around Mendota today. I've visited a few places where I think he might have collected from in the early 1900s, some sphagnum. I think it might have been Cherokee Marsh, which is where I grew up, and it also might have been Pheasant Branch Conservancy. Those are both histosol soils, so that might have possibly been where it was collected at the time, or maybe it was near the UW hospital. But um, what I am confident is that it's not present there anymore. And you'll see that little cluster of collection sites in Barron, where sphagnum is present at 2 of those, and then up north in Bayfield County, those lighter pink those lighter pink dots um, indicate a Jacquard index value closer to 1, which would be identical data sets, but none of them quite made it to the identical data set that Cheney had collected, but the closest were in Drummond and at Port Wing in Bayfield County, Wisconsin. And so with that, I would like to start on section 3, the art and labor of identifying sphagnum moths. So I would like to take a moment to appreciate the diversity of species within the genus Sphagnum. I think a lot of people, when they think of sphagnum, and by a lot of people, I don't mean all of you necessarily in the Botanical Club of Wisconsin, but I think when a lot of people think of sphagnum, they think of those large, juicy hummocks that you'll find in an open bog or maybe in a conifer swamp. Um, but what I've really come to appreciate about this genus over the past year is the diversity of color, of shape, of size, of growth form, and habitat. Um, and really just the unique personality of each of these species of sphagnum. They are just so distinguished from each other. They're so unique. Um, there are 25 or 29 species of sphagnum known to occur across Wisconsin, and I think it's really worth getting to know each and one of these species, even if they are pretty difficult to tell apart from each other in the field sometimes. But they can tell you a lot about the ecology of the site that they are present at, as we have learned about in this presentation. So some field characters that are really useful to know in the field to get to know the specimen that you are looking at. In addition to habitat and biotope, obviously, you're going to want to look at the capitulum, which is the head of the sphagnum. Is it flat? That might be sphagnum phalanx. Is it more convex? It might be sphagnum capillifolium. You're going to want to look at the size. Is it a really large for sphagnum, or is it pretty small? And the branch organization, if you've got recurved branches around the top of the capitulum, you might be looking at something within the subgenus Subsecunda. You're going to want to look at the terminal bud. Is it conspicuous or inconspicuous? This can cross off a lot of species that you might be suspicious of. You're going to want to look at the number of branches per fascicle. If you are in Wisconsin and you notice that there are 6 plus branches on a fascicle of your sphagnum moss, it might be, well, it's definitely Sphagnum wolffianum, because that's the only species with that many branches packed in there. If there's 2 spreading branches and 3 pendant branches, that could be Sphagnum teres. But again, these characters are meant to be in use in combination with each other. None of these are going to be really a 100% giveaway for one species most of the time. You're going to want to look at the plant and stem color, and while this is often variable within a species and even within an individual, it can help you identify it to subgenus at least. So is it red? Is it brown, green, yellow, blackish, etc.? You're going to want to look at the stem leaves, and this can be challenging in the field, but if you have a hand lens and you rip off that capitulum, you can sometimes hold it up to the light and you can see the general shape, size, and the apex of that leaf. And so you can get in a good idea if that apex is really eroded. Maybe you're looking at Sphagnum fimbriatum. The branch leaves, if they're cuculate, which means spoon-shaped or easy to count with the naked eye, you might be looking at Sphagnum subgenus Sphagnum. If they are extremely squarose, you might be looking at something within the subgenus of Squarosa. And the presence or absence of sporophytes is something to note definitely in the field. Some you might be between 2 species that look really similar, and if it has a sporophyte present, for example, Sphagnum fimbriatum really commonly occurs with sporophytes in the field, at least in the spring. But Sphagnum gargansonia, a species that looks really similar to it, is rarely found with sporophytes in the field. So that might give you a better idea of which species you're looking at. But with that said, it's really, really, really difficult to get to species in the field, and dare I say, impossible. I mean, if you are an extremely seasoned sphagnologist and you've been doing this for many years, you might get a really, really good guess of what you're looking at in the field. But you're never going to be 100% right 100% of the time. I promise you that. And especially when you are a beginner and you're just beginning to learn these species, you're definitely going to want to get to know them under a microscope. And so I want to tell you about some sphagnum microscopy supplies to get started if you're interested. You're going to want a compound scope and a dissecting microscope. Obviously, you're going to want microscope slides and cover glass, an assortment of high quality, and I stress that high quality. You're not going to want to be pinching some tiny 1 millimeter long leaves with some, poor quality tweezers. Some crystal violet dye. This is useful for dyeing the leaves of the sphagnum. That highlights really important characters like porosity and fibrils that you cannot see in the field. Sharp, thin razor blades for making cross sections, distilled water and a dropper, beakers and sacrificed dishes for that crystal violet dye, and a really good book. So I can make some really good recommendations for these books. On the left here, we have Sphagnum mosses of Eastern Canada. This is an incredibly good book. It's taught me much of what I know about sphagnum. It's usually the first one that I grab for. And on the right here, Sphagnum Moss's The Stars of European Myres. This is an incredibly comprehensive book, and what a great name. Both really great resources. And so the first step to sphagnum microscopy back at the lab is going to be to rehydrate your specimen. So maybe this is a little less important if you just collected it that day and it's still wet, but especially for those collections that have been drying in the herbarium or at your house for many months or many years, you're going to want to rehydrate it. Now, there is a shelf life to some of these specimens, and most of those that were collected by Cheney so many years ago are no longer fit to be rehydrated. They just kind of turn to mush, unfortunately. But uh, for our sakes, for our intents and purposes, you're going to want to rehydrate your specimen, and then you're going to want to put it under a dissecting scope and really get to know it here. This is usually where I start to take pictures. I usually take pictures to upload to iNaturalist observations, or sometimes, or most of the time, I include a lot of pictures of all the diagnostic characters under the scope with my collections that I upload to the Bryophyte database. Step 3 is going to be to remove all of those fascicles along the stem. And so I'd like to point out one fascicle of sphagnum over in the top right corner. So that's got two spreading branches, and those are those branches that travel perpendicular to the stem a bit and then taper it off down. And then it's got, I think it's pretty hard to see, but there's three pendant branches on that fascicle. And so those pendant branches are going to be those that are more oppressed to the stem. And so you're going to want to note how many of those spreading and pendant branches you've got there, and then take those all off and then drop the stem and one of those removed spreading branches into crystal violet dye. And then after they've been sitting in that crystal violet dye, give it a little rinse with some water. And then step 4 is going to be to prepare a slide of dyed stem leaves. And so under the dissecting scope, you're going to want to note the length of that leaf. That's often really important. And then under the compound scope, noting things like leaf length and, sorry, you hear a train. I live on University, so it's quite loud. You're going to want to note stem leaf shape and cell porosity and fiber pattern. And so, again, I would like to take a moment to appreciate the diversity of stem leaves within this genus. This is often the character that I turn to first if I have absolutely no idea what I'm looking at, and often the character that I'm most confident in allowing to tell me what species I'm looking at. They are really just so diverse across species. You've got, such different apexes here. You've got this really pointy one, Sphagnum phalanx on the first slide, and then this more rounded one, and then a much more fimbriate one, that's Sphagnum fimbriatum. This is a really, really great character to look to. Step 5 is going to be to prepare a slide of branch leaves. And so I'd like to draw your attention to this little diagram of a branch here, this picture of a branch. And I've got an arrow pointing to one itty bitty teensy little a branch leaf there. And so you're going to want to carefully remove those with your high quality tweezers and toss them on a slide. And then under the compound scope, you're going to want to note things like the shape overall of the leaf and cell porosity, fibro pattern, et cetera, et cetera. In the bottom right-hand corner, I'd like to point out those little white spaces within the leaf, and those are pores. And that is something that is really important oftentimes to look at on a branch leaf and can tell you a bit about at least the subgenus that you're looking at, if not the species. And again, that's something you can't see in the field. So some additional preparations that you may or may not make. Branch leaf cross sections. This is helpful to get to subgenus and often necessary for identifying within the subgenus Sphagnum to species. So you're not going to have to do this every single time you collect a specimen, and it's pretty difficult, but once you get some experience, it gets a bit easier. But really what you're looking at here is the arrangement of the hyaline cells, which are those big inflated clear cells, in comparison to the photosynthetic cells that are green in between them. So on the top right, that is Sphagnum palustri. You can see that those green cells are a bit triangular shaped and wider on the bottom than they are on the top. And then on the bottom there, you've got Sphagnum papillosum, And you can see those photosynthetic cells, they're pretty papillose. You can see those little protrusions. Some additional preparations you're going to want to make, but not every time. Stem cross section. This is important for identifying the species within the section uh, subsecunda. So all the way on the right there, you've got you're going to want to look at those cortical cells and how many layers there are present in your specimen. So there's, I think, 3 or 4 present on the far right. In the middle, you've only got 1 layer of cortical cells, and then on the left, you've got maybe 2 or 3. So this is often a really important diagnostic character. And finally, you've got stem cortical cells. It died under a microscope, and here you're generally looking for porosity, and this can tell you the difference between 2 closely related species. This is often kind of just a supplementary character, but sometimes it's pretty useful if you're in between two and you're not really quite sure what they are. And so, with that, I would like to say thank you, thank you, thank you, thank you to the Botanical Club of Wisconsin for your support on this project and for the funding that made this possible. Thank you to each and every single one of you that came out tonight to listen to my presentation. I really, really appreciate it. And to the Wisconsin State Herbarium for employing me and for giving me so many opportunities that have brought me to where I am today. To Dr. Marianne Weiss, my boss and my mentor, and her love for bryophytes is what made this project possible. And I've learned so much from her, not just about bryophytes and about herbaria, but also just about general botany. She's a wealth of information. Paul Helena, thank you very much for your support in Barron County and for letting us borrow your canoes and for being the best company, the best, funniest company I've ever had on a canoe. To Eric Sorensen for joining me on so many of these trips and for always being down for an adventure, even if it's, pretty clogged with cattails and poison parsnip, and for listening to hours and hours of me talking about Cheney and sphagnum. To Mike Krolwitz and Brandon Quarter, dear friends of mine, as well as coworkers and mentors who have endlessly inspired me and taught me so much of what I need to know about botany. And to Brenda, who joined us in Barron County and is always a pleasure, to the Peatland crew at the herbarium, and finally, last but not least, to Leland S. Cheney for his contribution to the flora of Wisconsin, both great and small.
Awesome. Well, thank you, Holly. That was really great. A really cool mix of sort of like historical research and practical lab techniques, but then also the floristic work, which was really interesting. Okay, so if people have questions, we have a little bit of time, so put them in the chat first, and we'll go through them there. If for whatever reason you can't access the chat, or you think your question would be easier to just say out loud, feel free to raise your hand, and then we'll call on you, and you can unmute yourself. Um, so yeah, so as maybe we'll give people some time to write down some questions. I know I had 1 or 2 things. One thing I was thinking of, Holly, when you said your at the beginning, you had a comment about like the importance of writing um, precise descriptions of
Mhm.
locations. Whenever I think of L.S. Cheney, I think of his he has a number of specimens that just say Lake Superior region or Lake Superior area. I always think that's funny. That is totally unhelpful.
Yeah, I think he used that label as a general label for all of what he collected in like one season when he did what he called, one survey of the Lake Superior region. And so if you go into
Yeah.
those collector's logs, you can find more
Ah,
specific locality information, but a lot of people don't know that if they haven't spent, 2 years working with those
right.
collections. And so they'll just put what is on his label that I was talking about, collected and identified by Leland S. Cheney, and a lot of the times it says in the Great Superior or in the Lake Superior region.
Yeah.
Um, but
Oh,
that's
that's
pretty
interesting that
pretty
there's
broad.
yeah, right.
Yeah.
Yeah, I didn't think about these logs. Oh man. Okay. Questions are pouring in. So I'm gonna, uh, shut up and get to these. Okay. How many species have been split, described since Cheney ID'd them, and could that contribute to some different IDs between the original collections and resampling?
Yeah, so a lot of the species actually within sphagnum are still pretty relevant today compared to when he was doing his identifications back in the 30s. There are some species complexes that I indicated in throughout this presentation, that being Sphagnum magellanicum complex. That's a group that we're still working on. John Shaw does a lot of work within that group, and you might have heard me referring to anything that was labeled Magellanicum complex as divinum, um, just because if it's red and it's within Sphagnum subgenus Sphagnum, it's probably divinum because that's the only one that occurs in Wisconsin. But across the globe, there are several species in that complex that are constantly being rearranged. And another one is the recurvum group. Unfortunately, I think in the 60s we had a few collectors or a few botanists that went through and annotated a few specimens of Cheney's, I think before they got all mushy and impossible to identify. And so I trust those identifications within the recurvum group. But a lot of the other ones that were identified by him, like he would say Sphagnum angustifolium, uh, I wouldn't necessarily trust that, even though that's still a relevant species today, just because that has been split into a few other species that we do find around Wisconsin pretty commonly. Um, so generally, I trust them, but there's a few complexes that are a bit more complicated.
Okay. Glenn asks, I live near Cedarburg Bog. There's lots of tamarack there. Is there sphagnum there too?
Yeah, definitely. And I've never had the pleasure of visiting yet. Marianne might want to tune in a little bit on this. Um, because we might team up with Cedarburg Bog a little bit into the future with this peatland crew that I was talking about. But yeah, there's definitely sphagnum present there, and Cheney never collected at Cedarburg Bog in particular that I know of. But yeah, I would love to make it there.
You said you've never had the pleasure of going there. You might that word might be used loosely. There's a lot of poison sumac.
Oh boy. Yeah.
Uh,
Marianne, what did you get us into?
Yeah. So we will be going there this summer. But yeah, there is lots of different sphagnum there. And so, yeah, so that's part of the reason we're going. But yeah, we're going to be doing a project with Cedarburg Bog this summer. So yeah, more to come.
All right. Next question. How do you confirm? You sort of got to this one already, Holly, but Michael asks, how do you confirm or correct any of the IDs of Cheney's original names since you can't rehydrate the specimens?
Yeah, so generally I trust them if they've been annotated somewhat recently by, like Warnsdorf. You might have heard me referring to Sphagnum Warnsdorfii, which is named after him. He was one of the world's most renowned sphagnologists, and he came through and annotated a few. I don't know if he annotated them at the herbarium, but he annotated a few that we have at the Wisconsin State Herbarium. And so I'll trust specimens that have been annotated like that somewhat recently. Um, and yeah, sometimes I just don't trust them if they're in a complex and treat it for what it is, which is within the complex.
Yeah, one thing about that, if I can just add, is that a lot of Cheney's sphagnum specimens he actually sent off to this Leroy Andrews who was in out at Cornell, and so he actually sent almost all of his sphagnum specimens. And this Leroy Andrews, he was a sphagnologist, an expert in sphagnum, and so he identified a lot of his. So yeah, so we can we trust. I guess we trust that quite a bit. But Dick Andrus also, who he came by in the I think maybe the 70s and checked some as well. So we're fairly confident in most of the IDs. Yeah.
Yeah, and I think about that a lot because Cheney has like over 400 specimens that we have entered in the herbarium. Um, and there might have been even more that we don't have yet or may have been lost over time. And so I wonder if he sent like 400 specimens to Andrews to identify because it took me a long time to get through the 120 that I collected last season. So that must have been quite a project.
Okay, Derek asks, were any of the Barron County sites near Cumberland, which is in the northwest part of the county? There are more intact peatlands within the county forest there. And then Derek says, if you need any help finding sites in northwest Wisconsin like Burnett or Barron County, let him know. He's glad to help getting access to those sites.
Thanks, Derek. I appreciate that. Yeah, I know Marianne, when we went up north, she went and she visited a few more intact bogs around that area. Marianne, did you make it near Cumberland? I'm not sure where Barron, the city, the town is in uh in comparison to Cumberland.
No,
This is Derek. I think I crisscrossed paths and didn't get to actually meet you. And when we went
Oh,
out with Mary Ann, I went out with Mary Ann either before or after you came up to Barron County, and we went to some places, but not near Cumberland. So Barron's
oh,
kind of in the
sure.
center of the county, and Cumberland is in the northwest corner of the county. But there's a lot, like I said, there's lots of peatlands within the county forest up there that are small little pocket wetlands and things that might be worth visiting. Um, as far as intact systems go. So
Gotcha. Thank you for that. Yeah, I'd love to make another trip up there. Definitely. Put that on the list.
And then someone, I think this might be Paul Helena, asks, are you going to continue your grad studies with bryophytes and which, if so, which groups?
Asking the tough questions here. Yeah, that's the plan. I'll be working in the Hotchkiss lab with Sarah Hotchkiss here at UW, and I'll be coordinating with Marianne. I'll be sticking around here as well, and the plan is to maybe stay with Sphagnum. Sphagnum and I are not married. I'm open to learning about more plants, but I have really fallen in love with this group over this project, and so I would love to continue on with it for the next couple of years. So it's in the
Cool.
works. The project is in the works. I'm not completely sure yet.
Sounds good. Let's see. Michael asks, in places where sphagnum has disappeared despite fairly good habitat, what are the possible causes? And he lists maybe water table. The water tables are compromised by nearby buildings or development. Maybe some other
Yeah,
things.
definitely development. I talked about a site that was collected at by Cheney and it was now a Ace Hardware parking lot. So development's one of them. I think that draining and ditching is definitely a leading cause in across Wisconsin. Just rerouting and adjusting that hydrology, and it's making it way drier. And sphagnum needs a relatively high water table in order to uh, persist. So I think that hydrology is a really main factor, something that not a lot of people talk about in the literature, but something that I think really played into that encroachment of minotrophic species in Barron County is nutrient flow. And so that kind of ties into hydrology, but I think that maybe nutrient inputs onto cropland might be facilitating the persistence of more minotrophic species and less ombrotrophic species that are a bit more restricted to less nutrient-rich sites.
Did you look at any of the historical aerial photos for any of those sites, like from the Wisconsin Historical Society? They have those 1937 aerials.
Yeah, yeah, we've got some of those at the Wisconsin State Herbarium, and I did look over them. And sorry, I meant to talk about this a bit in my presentation. Interestingly, those go back to like the late 30s, at the earliest, and most of his collections were before then, and so I think they're really useful for seeing a snapshot of what they looked like then. But um, for example, in that section 20 where I found those species amongst the alder thicket and in the cattails in Barron, Wisconsin, I expected, oh, that's interesting. I wonder if, more recent, like after Cheney collected, if farmland moved in and then the leaching of the nutrients off of the land, choked that sphagnum out, or, they just plowed over peatlands and, drained and dredged and made it into farmland. But that wasn't the case. As of 1939, it was already a cropland. So I'm not sure what the state of that site looked like in 1893 when
Hmm.
he collected there. I know that settlers first settled in Barron, Wisconsin, along that Quadra Creek actually in 1860. So that gives about 30 years to establish some farmland. I don't know if they had plowed over that sphagnum habitat at that point. But what I do know is that over the last 130 years, it's made way for the persistence of much more monotropic species.
Yeah, interesting. Okay. I think Paul Helena asks, will you travel north to do more workshops in the 1896 Cheney Lake Superior region? Looking for dates in May or June, mostly the Duluth, Minnesota area. So yeah, are you going
Am
back up to the
I
Lake
going
Superior
back
region?
up? Well, definitely. I'm not sure if it will be specifically for this Cheney project. I think um, most of the sites that I visited in northern Wisconsin were the only
This-
ones that had
Can
specific
you
enough locality information. Oh,
hear me? This is Paul.
hi,
Yeah.
Paul.
Yeah, that was my
Hey.
question. Yeah, we have a native plant group, Native Plant Explorer, and I'm not on the board, but I'm a frequent participant. And they asked me to ask you if you would come up, and we have a few dollars for you to come up to a May or June meeting, and if we can actually tie it with a sphagnum moth, I don't know, there's not that much in Duluth, but maybe Eloise Bay in Wisconsin might have a few sphagnum. We'd be willing to pay some. We don't have a lot of money, but we might have be able to entertain having you up. I loved your presentation. It was very informative. Like Kevin mentioned, I'd love to know what the historical land was going on throughout each of your study sites. And it's pretty obvious that the Bayfield County area, which is least developed and would have the greatest diversity of sphagnum, as it does the greatest diversity of other vascular plants as well. So anyway, if you want to just send me an email and
Yeah,
see if you'd entertain that, probably May or June, because we don't have much time in April, so it's already April. So um, that was the
yeah.
question I had for you. Okay.
Okay, absolutely. Yeah. We'll be in touch.
Okay.
That sounds like a lot of fun.
Awesome. Okay, three more questions and then we'll cut it off there. What is the effect of salt runoff on sphagnum?
That's interesting. I'm not completely sure. I mean, there were a few species that we collected not too far from a road, so I'm not completely sure that it doesn't allow them to grow, but that's a good question. I would like to look into that.
Let's see. Chris asks, beaver and any other artificial impoundment, especially in the presence of some kind of urbanization or agriculture, can mess up a low nutrient sphagnum supporting wetland for a very long time. Okay, so I guess it's not a question, more of a statement.
Yeah, absolutely.
Yeah. And then Brenda asks, any sphagnum restoration projects going on in Wisconsin that you know of?
Yeah, I would like to look into that more. I'm familiar with a few efforts, I think, on campus. I have a friend who says she has some friends who are trying to figure out what species of sphagnum would be the best for restoration purposes, and they're going to try and run some experiments on that. It's something that I'd be interested in getting involved with. Um, yeah, but in terms of sphagnum specific restoration, I think it's pretty slim. How about
Yeah.
you, Marianne? Do you know of any?
No, I don't know of any in Wisconsin offhand. No.
Yeah, that's like next level wetland restoration is restoring
Yeah.
peatlands. But I'm sure it'll be on people's minds soon.
Mhm.
Okay. Well, yeah, we'll cut the questions there. Thanks again, Holly. This was really great, and I think a lot of people really enjoyed it, as shown by the attendance and all the questions. So people were really engaged. So again, thanks a lot. Um, Glad to see you could do a lot with the Botanical Club Thompson Award. Um, and yeah, thanks for giving this presentation. It was great.
Thank you guys again. Thank you for everyone who showed up. Appreciate it.
Yeah. Okay. Well, yeah, so that's it for this season of presentations, everyone.